Welcome everyone to a new episode of Genuine Rockstars, and today's Genuine Rockstar is Hannah Byrne. How are you, Hannah?
I'm good. Pleasure to be here.
Could you tell our audience a little bit about who you are and what you do?
Yeah, of course. Yeah, so I am a PhD student at Uppsala University alongside yourself. However, I am soon to be defending my PhD. My PhD looks at environmental and ecological aspects of what is known as the fish-to-tetrapod transition. So specifically, I look at whether large tides could have been a driver in fish moving out onto the land. And I also look at tetrapod body fossils from just before a mass extinction event. And I also look at fossil poop, which I have to say is my favorite part of my studies.
So you're a paleontologist with a specialization in early tetrapods and in particular in their poop. What got you interested in paleontology in the first place, and how did you end up studying fossil poop?
Yeah, well, it was quite a journey. I didn't start out at the age of 3 or 4 with aspirations to study fossil poop. My background actually is in marine biology and oceanography. During my degree, I was looking at internal tidal waves and how they upwell this nutrient-rich but also cold water to the Pacific reefs and islands and makes them very productive. So it was actually a talk given by a professor, Steve Balbus from the University of Oxford, who's an astrophysicist. And he's had a fascination on why we have life on Earth. And he was particularly focused upon the gravitational forcing of the moon and the sun and Earth. So he looked at the Devonian period and thought, well, this is a crucial point in the evolution of life on Earth. And he thought that large tides during this time could have been a big driver in fish moving onto land. So I was at that talk, I was hooked, I got involved in the project, and the rest is history. I ended up getting really fascinated with tetrapods, and yeah.
Our audience may not be familiar with the whole concept of early tetrapods. To give them an example, we are late tetrapods, today's tetrapods. So what are early tetrapods? Could you maybe explain that?
So the fish to tetrapod transition refers to the transition of fish onto land. So this is when we have the first vertebrates, so animals with a backbone, moving onto land. So this took a period of time. So we start off with fish known as lobe-finned fish, and they go through a series of evolutionary events, and they acquire limbs and lungs, which of course are very important if you want to, live on the land. And the first lobe-finned fish that we know of dates back to around 425 million years ago. And then the first evidence that we have of the first tetrapods walking competently on the land is around 390 million years ago.
You recently published on coprolites from East Greenland. First of all, wow. From the beginning of the Carboniferous period. Could you tell us what you studied and what you actually discovered?
So the material comes from a lake deposit from East Greenland. And as you mentioned, this is early Carboniferous. In fact, it's probably the earliest Carboniferous deposit we have. And it's also our first insight into an ecosystem immediately after the Hannah extinction event.
The end Devonian extinction we can sort of put into 2 waves, but the last one I believe is the real shebang, and it's called the Hannah crisis.
So we have the Kael'Vasser event, which is 10 million years older than the Hannah event, and the Kael'Vasser event predominantly affected marine ecosystems. However, with the Hannah crisis, it's impacting both marine and terrestrial ecosystems during a 300,000 year period. And it results in a over 50% decrease in vertebrate taxa. And we also have the loss of one of the major fish groups of the time, the placoderms. So in this deposit, we have a large collection of body fossils belonging to 3 different types of fish. So from this deposit, we're getting a perception that this is a low diversity fauna. So maybe something we would typically expect in a recovery fauna after such a major extinction event. But we do have a big fossil poop or coprolite collection also. So we have over 200 fossils. So that was my job, basically, to analyze this collection to see what can the coprolites tell us, and are we missing any kind of fundamental pieces of information?
So you nuke them with X-rays and you look inside?
Yes. So for this project, we did a combination of examining the external features, so the shape and size, but combining that with the brilliance that is the scanning to be able to look internally, to get like a really comprehensive idea of, okay, what are these coprolites? What's in them? Are there distinct groups? And what is this telling us?
Yeah. So just for our audience, if you look behind Hannah, you see the cross sections of the scanning of her coprolites. So she has a wall,
yes,
and she threw a lot of shit up against it.
Literally, yes. I had it described by a colleague of the wall of a psychopath. But yeah, so basically these are virtual thin sections that we obtained from the scanning. This is showing us, what's inside the coprolite, what's the kind of matrix made of, is there any shape to it? So it was nice to have it all kind of together for my kind of easy visualization.
What are the most exciting things you found inside coprolites?
What we found is that we have 8 very distinct groups of coprolites. What this told us was that we're not fully capturing the diversity of the ecosystem of this locality. We only have 3 different types of fish from the body fossil record, but clearly we have a kind of hidden diversity represented in the coprolites. One of the primary things that you want to be looking for in terms of the shape of coprolite is whether they're spiral or non-spiral. So spiral will indicate whether the animal had a spiral gut valve. So in present day, that's in sharks and lungfish and some primitive ray fin fish. But during the Carboniferous period, most animals had a spiral gut valve. However, one group that did not have a spiral gut valve are the tetrapods. And finding these large non-spiral coprolites indicates that we had tetrapods in our ecosystem.
So even though you've never found any body fossils of tetrapods there, you find their shit, exactly. So they were there.
This is important for this time period because we have a period what is known as Roemer's Gap. And Roemer's Gap was a concept developed by Alfred Roemer in the 50s, which he thought that there was this 20 million year gap between tetrapod fossils from the Devonian and tetrapod fossils in the Carboniferous.
That coincided with the end Devonian mass extinction.
The fossil evidence at the time showed that there was this gap. And so there was this perception that the late Devonian mass extinctions, had a catastrophic impact on tetrapods. But research over the last kind of 2 decades is starting to fill in that gap. Very recent discoveries in Greenland, combined with the evidence of tetrapods from the coprolites, suggest that, there isn't a gap. It's more of a sampling issue or sampling bias.
If you don't manage to look inside the rocks of those ages, then you're never going to get stuff from that time.
As paleontologists, we know that having things preserved is really special. It's very rare.
You're almost done with your PhD at the time of this interview. And by the time we release this video, you're a doctor. What are your short-term plans? Where do you see yourself a few years from now?
Okay, so in the short term, I will be continuing on some of my PhD projects. So I will be writing up a paper on the virtual reconstructions of the contents, which you have helped me out with. So that's so that would be a very exciting paper. And yeah, beyond that, I don't have major grand plans.
So which advice do you have for our viewers who also would like to pursue a research career in vertebrate paleontology?
Absolutely go for it. You don't have to have had a burning desire for dinosaurs from the age of 3. Like I come from a marine biology oceanography background, and I think what I didn't appreciate about paleontology until I started to get into it is how diverse it is. There are so many geological periods, there are so many fossil sets, so many deposits, and so many different aspects. You can look at, plants, you can look at spores, you can look at invertebrates, you can look at poop. So there really is something, something for everyone. And don't be afraid to reach out to people, go to conferences, make contacts. And, it will be tough at points, but it's a really enjoyable research field. It's a very rewarding research field. So.
And this is coming from the lady who studied shit.
Exactly. It's a shit job, but someone's got to do it.
So how do you like to spend the hours during which you're not studying fossil poop or sleeping?
So I took up climbing actually in early 2020. So that's kind of been my new obsession, hobby, passion. So I'm very fortunate that there's a lot of nice outdoor climbing here in Sweden as well. So I spend a lot of time indoor climbing, outdoor climbing. Yeah, I also like to do meditation. Try and do that every day. But I also, I mean, coming from one of the reasons why, the main reason why I went into marine biology and oceanography is that I love the sea. I love the water. So I'm a massive water sports junkie. In general, being outdoors, being active.
Thank you so much for talking to us today, Hannah. You're a genuine rock star.
Awesome. Absolute pleasure. Thank you.